Okay, our final speaker in this section is Rebecca Buchanan. Rebecca studies the migration of salmon and steelhead from the statistical viewpoint. She specializes in designing and implementing release capture models to estimate migration routing, and survival in complex river systems, including the San Joaquin Sacramento Delta. She's a senior research scientist in the School of Aquatic and Fishery Science at the University of Washington in Seattle. She received her BA at Bryn Mawr. See, if I was back east, I would know that. In math, a Master of Science at Syracuse University in math education, and in 2005, her PhD at the University of Washington in quantitative ecology and resource management. Rebecca?
All right, hello, and thank you for the opportunity to speak to you today. I'll be talking about a couple of large-scale acoustic tag studies from the San Joaquin River through the Delta, looking at juvenile salmonid survival. Oops, there we are. So I'll give a quick overview of these studies and then talk about some of the issues we had with just analyzing the data because of the predation by predatory fish. And then go to the results. That's for fall Chinook salmon from 2010 and 2011, and steelhead from 2011. And the steelhead results are brand new. They're preliminary results. We're working on the report right now. And then some conclusions, and then if I have time, just one slide on some future and ongoing studies. Right, so the studies that I'm talking about are these large-scale South Delta acoustic tagging studies, and we were looking at survival from this region of the San Joaquin, so around Mossdale Bridge, through the Delta to Chips Island. The studies were the Vernalis Adaptive Management Program tagging study, or VAMP. This was a multi-year study, 12-year study. And I'm just going to talk about the last 2 years of data. It ended in 2011. So I'll talk about 2010 and 2011. The previous 10 years, we didn't have acoustic tag data throughout that entire time. You really only got some really data of the quality that we wanted in the last 2 years. So I'll talk about that. And the South Delta Temporary Barriers Study is done to look at the passage past the temporary agricultural barriers that Jacob talked about earlier. But we can use the release groups from that study to study survival through the Delta. And then the 6-year study, or 6-year steelhead study, began in 2011. So I just have the 1 year of data from that. So these were studies that were led and implemented by the U.S. Fish and Wildlife Service, Bureau of Reclamation, Geological Survey, California Department of Water Resources, and the San Joaquin River Group Authority. Right, so here's another map. And this map I'm going to be using as the basis for a lot of my slides. So we're looking at survival from Mossdale to Chips Island through any of the routes that the fish may take. Here we have the water export facilities, the federal facility here, the state water project here, Clifton Court forebay here. We had receivers at the entrance of the Clifton Court forebay at the radial gates, but we did not have receivers throughout the forebay or in the actual water project itself. And I'll show you where the other receivers were. The distance that we're looking at, the travel distance for fish that went through the San Joaquin was about 91 river kilometers. Of course, going through other routes would be a different length. So there are lots of routes the fish could take. I've just color-coded some of them here, including the route that takes fish over to the export facilities, and then if they're salvaged, they get put in a truck, and they're trucked down to release sites down in either the San Joaquin or the Sacramento, where they're released and then they can go on out. So the route-specific survival for Old River includes that trucking route. So I'll be talking about two primary routes, and those are defined by what happens at the head of Old River. If they stay in the San Joaquin here, then they're in the San Joaquin route. If they enter Old River, then they're in the Old River route, and that would include the possibility of exiting Old River at Middle River and then getting over to this region, either going downstream or through the delta on their own or going over to the facilities and being transported. So we tagged between about 1,000 and 2,000 or so juvenile Chinook and or steelhead in both of these years with HDI microacoustic tags. We released them down here primarily at Durham Ferry on the San Joaquin River, and then we monitored their migration using fixed site receivers, which are denoted by the red bars here. So we could follow their migration through the delta. And the receivers were placed so that we could estimate reach-specific survivals and also route entrainment probabilities at key river junctions, such as the head of Old River. So we analyzed the data in a release recapture model that was designed specifically for this system. This is a schematic of the model for Chinook from 2011. And I'm not going to go through it except to point out that it's just modeling the paths that the fish may take. And we're estimating from that reach survival probabilities, route entrainment probabilities, so that's the route use, and transition probabilities just combines reach survival and route entrainment, and then detection probabilities. So we fit the model in software designed for these types of models, developed at the University of Washington, and we got our estimates. So if that was all it was, it'd be kind of simple to fit, not super simple, but not too complicated. The problem was that with the predatory fish, if they're mobile predatory fish, they eat our fish, then they're temporarily tagged, so they go past a detection site, and then we get a detection. And as Jacob was describing, we have to try to identify which of these detections came from predators and which came from our study fish. So our method was to develop a predator filter. It's a deterministic filter rather than a probabilistic filter. And it's based, again, on assumed behavioral differences between our study fish and the predators. But instead of using more of the fine scale movements like Jacob was talking about, we're using movements throughout the entire array of our receivers. So it's a much larger scale analysis, more of the far field and midfield scales. So we were looking at the residence time of a tag in the vicinity of the receiver, migration rate between receivers, movements between receivers. Are we seeing fish going upstream? Are they cycling through the delta? Total travel time from release, and then we were analyzing this in the context of river discharge, water velocity, and tidal cycle, primarily looking for movements against the flow. So this was a spatially explicit rule that had different criteria in different parts of the delta, and it was developed based on conversations with biologists that are familiar with the region, familiar with the fish here. So the behavioral assumptions, first, salmon smolts, we're assuming we're actively migrating downriver. They're unlikely to move against the flow or to linger in a given area unless they were experiencing reverse flow, which happens quite a lot in this region, in which case we allowed them to have more flexible behavior. For steelhead, it was a little more complicated. We basically just had the same assumptions as for salmon smolts, but we relaxed them some. We know that the steelhead may rear in the deltas. They may look like a predatory fish. They may be a predatory fish. But we are assuming that if they've entered the delta, so in particular if they've gone past the head of Old River, that they were actually actively migrating. Maybe not very quickly, but we're assuming that they want to go out unless they're eaten. Above that region, we couldn't really say if they were actually a predator or maybe they were residualizing. For the predatory fish, our model for this was the striped bass. We know that doesn't include all predators, but we thought that would be a reasonable model for most of the mobile predatory fish, and those are the ones who are really the problem. So we assume that they may move against the flow, they may linger in a given area, and they may move either very quickly or very slowly between detection sites, may also cycle, either on a small scale or on a large scale. So using this filter, we then removed the detections that we thought came from predators and then fit the model. So I'm going to show you some results. And I'm going to focus on trying to identify where the predation is primarily occurring. So we have two ways of identifying this using the data that I've been talking about. First is the predator filter results. Where does the predator filter say that its predation is happening? And then also we've got the reach survival estimates. And that's based on the output of the predator filter results, but it would also capture predation by other types of predation that the filter would not identify. And then I'll also show you estimates of route-specific survival, route entrainment, total survival, and travel time. So we'll start with 2010 fall Chinook salmon. These were released in late April to late May of 2010. Their average fork length was 110 millimeters, and these were hatchery fish. Right, so most of the results slides are going to look something like this. The underlying map here is the same map I was showing you before. I just color-coded it blue to have something that wasn't white. So it's not saying that it's all water. It's just a nice color. The discs that are shown here are covering the different regions of receivers, and they're showing you the numbers of tag detections at each of the receivers. So that includes the fish that we thought were, or the tags we thought were in predators and the tags we thought were in salmon smolts. Then the shading of the discs indicates the percentage of those numbers that were first classified as in predators at that location. So there may have been other detections that had been previously classified as predators, but this is like the first time they were classified. So that's giving you one measure of the predation risk, either in that area or leading up to that area. All right, so for 2010 Chinook, we released 993 fish. Most of them were released down at Durham Ferry. We also had some fish released around Stockton and some fish released in Old River just downstream of the head. You can see that the numbers of detections tend to go down as you move down through the system, except for the supplemental releases, jacked it up again a bit. The shading, with the dark red indicating more predation than the lighter colors, the shading indicates that the predation risk was primarily in this region. So the central delta, this up here is where Turner Cut takes off from the San Joaquin, but it includes receivers on both the San Joaquin and in Turner Cut. We have a very dark disk here in Middle River, but we only had 8 fish detected there at all. Between 5 and 6 of them were first classified as predators at that site, and it turns out the other 2 were also classified as predators. They were just predators someplace else. That's not shown here. And then we had somewhat moderate predation at the water export facilities, well, at least at the entrance to the forebay and at the CVP here and in Old River downstream of the facilities. So then another way of getting at where predation is occurring is looking at the reach survival estimates. And so rather than putting a bunch of numbers on the screen, I just decided to go with the color coding again. So the arrows here indicate the reaches, and the shading indicates the magnitude of the survival probability through that reach. And this is a conditional survival, so it's not cumulative. So starting here, we have good survival through that first reach from Durham Ferry down to the head of Old River. And so dark green is good, white is bad. And then as you go down to the San Joaquin, survival tends to get less, and there's actually no statistical reason for that because, as I say, this is not cumulative survival, it's conditional. And once fish get down to this region, which is Medford Island, getting down to chips, there were some that did that, but really not very many. Survival kind of plummeted right around there. And that's the second, or this is next to the lowest category. It's not the lowest category of survival. For fish going through the central delta, we have these white arrows. That's a survival of less than 10%, so that's very low. Another indication that we've got predation going on here. Now for fish that got to the radial gates, we didn't have a whole lot getting there. This light arrow here means that. And of those that got there, none of them actually went onto chips. For those that got to the Central Valley Project into the holding tank, we actually had pretty good survival from there onto chips. And of course, these dashed lines, these are being trucked. So they're actually being trucked to around this area, and then they're swimming on down. Now these arrows in this region, this is indicating that not a whole lot of fish actually got into the holding tank at the Central Valley Project. And it's not actually a survival probability, it's a transition probability, which means it's the joint probability of heading off in this direction and surviving there. So if fish did not get to the holding tank, either that's because they died or maybe they went to one of these other 3 places. So if we add up all of those 4 arrows, we get overall survival in this region, and that was actually very high. It was above 90%, but from that general region on down to Chips Island, it was very, very low. If we combine all of those little arrows together, we get the overall survival from Mossdale to Chips Island, and we can do that in the 2 different routes, so the San Joaquin route and the Old River route. And in the San Joaquin route, it was 4%. In the Old River route, it was 7%. Remember, this is over, about 91 river kilometers, or 57 miles. It's not a long distance. We had a little less than half of the fish staying in the San Joaquin River at the head of Old River, and a little bit more than half entering Old River. Combining all those together, we get the total survival from Mossdale to Chips Island of 5% in 2010. And then we've got travel time. In the San Joaquin route from Mossdale to Chips Island, the average was 5.5 days, ranged from 4 to 7 days. That was based on 9 fish. For in-river or the Old River fish, those that went under their own steam, we only had 1 fish that we observed doing that. That was 7.3 days for what that's worth. For fish that were transported from they were transported from down here, but the travel time from Mossdale to Chips Island was a little over 2 days. So somewhat fast if you're trucked, which is not surprising. All right, so we'll look at the same types of results for false Chinook from 2011. These were released in late May or mid-May to mid-June, and again, their average fork length was the same as in 2010, 111 millimeters. So we released a little under 2,000 fish, all down at Durham Ferry, and we have the detections here. They declines as you go downstream. And the shading, again, indicates the percentage of those detections first classified as predators at that region. And so again, we're getting relatively high predation rates in this region, the southwestern portion of the delta, or kind of the central delta here. None at, or at least not very many, at Middle River. Quite a few at the radial gates at the Clifton Court forebay. Looking at the reach-specific survivals, we had a similar pattern as in 2010 with not horrible survival in the San Joaquin, but it is going down as you go downstream until you get to Medford Island here. In 2011, we had some sites at Jersey Point and False River here, and the survival from Chips Island down to this region was abysmal. It was, I think, 8%. So that really caused a problem for entire survival through that route. Once fish got to Jersey Point, they seemed to do pretty well at getting to Chips Island. Part of that may be that I didn't know how to really identify predation in this region based on the available data. But most of the tags that got to Jersey Point ended up getting to Chips Island. Again, we see low survival through the middle, or from the central delta, low survival from the Clifton Court forebay to Chips Island, high survival from the CVP holding tank to Chips, but again, not a whole lot of fish actually took that route. If we combine all these in this area here, we have survival through this region. Again, it's pretty high, not as high as in 2010, and then we had very low survival from that region onto Chips Island. If we combine all of that, we get total survival of 0.02, so 2% from Mossdale to Chips Island in 2011, which was a wet year as opposed to 2010, which was an above normal year. And survival was a little higher in Old River than in the San Joaquin, but I would not say it's a biologically significant difference. Travel time ranged from about 2 days or so in the Old River route to about 5 or so days in the San Joaquin route. For steelhead in 2011, these were released. We had one release group in late March, and the others were released in May through June. I'm just going to pool them all together for this. The average fork length of these fish was 277 millimeters. So these are pretty big fish, much bigger than our Chinook. So we are going to see higher survival, which is encouraging until you remember how big the fish were. And again, these are preliminary results. So here we have the detections. We released a little under 2,200 fish down at Durham Ferry. We had a lot of detections early on, and actually throughout we had pretty good numbers of detections. These are actually shaded by the results of the predator filter, but we didn't have any variation, and we had very low perceived predation rates for Chinook, or for steelhead. And if we drill down into this first category of 0 to 25%, we can look at shading just within that category. And we see that still, even when we do that, most of these regions had very low rates of identified predation, at least based on our understanding of the predators in the steelhead and their differences in behavior. We had actually the most or the highest predation rate appeared to be in this first reach from Durham Ferry down to the head of Old River. And whether that's really predation or if it's that our steelhead were residualizing there, it's not really clear. There were some detections that really looked very obvious like predation, like they were here for a long time and then they went down maybe to here and then came all the way back. We didn't think that was what a steelhead would do, but we thought that might be what a predator would do. We had some predation between the head of Old River and the head of Middle River, and also over at the Central Valley Project. And for all of these white discs, it's not that we had no predation there, it seemed to be very low, at least based on the predator filter. For the reach survivals, we had fairly high reach survivals in the San Joaquin throughout, low here as we would expect from the results of the predator filter. Lowish in these regions, kind of mimics what we saw with Chinook, but the magnitudes are higher. And we're seeing that while we do have lower survival from the Central Delta to Jersey Point than, say, in this region of the San Joaquin, it is still higher than we saw for Chinook. Of course, these are much larger fish. For steelhead, we had quite a few fish get to the radial gates at the Clifton Court forebay, and we had pretty good survival of those fish to Chips Island. And we are assuming that they actually were salvaged. We didn't have receivers in the holding tank, but we didn't see any of those fish going through the river to get to Chips Island. So we're assuming that they went through the holding tank. And we didn't have as many at Central Valley Project, but we still had pretty good survival from the holding tank there. So combining arrows in this region, we have pretty good survival from the head of Middle River through this region, and then good survival from that region to Chips Island. Combining everything, we have a total survival from Mossdale to Chips Island of 54%, which is huge compared to Chinook. Of course, our fish were pretty huge too, so maybe it's not so surprising. For travel time, the average travel time in the San Joaquin was 5.6 days, and also in the Old River via transport, 5.5 days. For in-river, a little bit longer. But you can see that the range of travel time was actually fairly broad, went from 2 to 35 days. And that maybe because the fish are rearing in the delta as they go. Some of them went on through, some of them took their time. All right, so I'll just sum up. We've been looking at information on predation from the acoustic tag data, and we have information both from the tag detections that were classified as coming from predators and the predator filter, and that's a very deterministic method. And then we have the reach survival estimates from the release recapture model, and that's more of a probabilistic method. So here again are the slides with the results from the predator filter. And we're seeing that for Chinook, we have what appears to be higher predation in the Central Delta and at the water export facilities in both years. For steelhead, we don't have evidence of any major predation problems, at least for this year. From the release recapture model, we have, again, evidence of predation in the Central Delta and in between Medford Island and Jersey Point here. And what I didn't say before is that may be happening in the river, in the San Joaquin, or it may be happening in Frank's Tract. We didn't have any receivers between Medford Island and Jersey Point, so we couldn't say which route the fish were taking. We didn't have the same types of problems with Chinook. Some evidence of mortality there, but not as severe as for Chinook. Okay, so I'm going to just wrap up with one slide on ongoing and future studies that I was asked to talk about, as I think many of us were. So these large-scale studies from the San Joaquin through the Delta are continuing. At least the steelhead study is continuing. It's a 6-year study, and so what I've been showing you is just the first year. So we have 5 more years of results. For the fall Chinook, that was coming primarily from the VAMP study, and that's over, but we did continue with a couple of more years of tagging. I haven't done the analysis yet, so that's coming up. And we're hoping for funding for 2014. Last year, there was a 2012 stipulation study that was looking at survival from the San Joaquin around the Stockton and Turner Cut area into the Delta through the Old and Middle River corridors and relating that to exports, I believe. And so Kramer Fish Sciences, I think, is still in the analysis stage, and maybe Brad's going to talk about that. Yes, okay. And then there's the Sacramento River study to the San Francisco Bay that we've been hearing about, and that's with all different types of Chinook, and everyone seems to be involved in that. Future studies, we've heard about some of these already. We know that we have quite a lot of studies that have been done, but no one has really compiled all of the results into one place and done a meta-analysis. So that is being planned to do a retrospective analysis of those data. And that would be on juvenile survival, which of course is just an indirect assessment of predation, but I think that could be very helpful. The predator removal study we've heard about a couple of times already. And then we know that if we want to be able to determine how we need to manage the system, at least as far as water exports go, then we really need to know more about fish behavior in response to water exports and tides and how they react to different hydrological conditions. So the South Delta Salmon Research Collaborative work group is working on designing a study to look at this. And this is a work group that's been convened to examine salmon research in the South Delta and really focus on how salmon react to the different water exports and how we can manage the system better to improve salmon survival. So everything that's on this slide is being done by groups that are associated with this work group. So for this study, we would love to study fish behavior, but we know that where we want to study these fish, there's a lot of predation there. It's over in the Old River near the water export facilities. It's a very dynamic environment, and we know there's a lot of predation there. So we need a tag that's going to identify predation for us, because we can't use a predator filter that's based on behavior differences to study behavior. Just like, I was showing you travel time. Well, I was using travel time as a criterion in the predator filter. So, travel time is somewhat based on my understanding of travel time, or our understanding of travel time, and we realize that. So we are waiting on a tag that will tell us when it's been eaten, and there are some tags some prototype tags, or a couple of tags in development. One's in testing right now. It's very, very early days, so we're not sure if it's going to work. But we're hoping we can get to that in study maybe in a couple of years. All right, and so then just want to thank everyone who is involved. These were very large tagging studies. All I did was analyze the acoustic tag data, but everybody else implemented the studies and planned them. So there's a lot of groups there. And then for the predator filter, a bunch of us have been talking about how to interpret the data and how we might actually distinguish between a predator and a salmon or a steelhead. And then folks at the University of Washington that have been very helpful too. So if I have time, I'll take any questions?
One or two.
One or two?
We can always do follow-ups.
I have a question about your detection probabilities.
Yes.
Can you go back to your 2011 Chinook slide?
Where?
Sorry.
This one?
So what happened in the San Joaquin River? You go from 841 to 473. What happened in there?
We had pretty good detection probabilities at all of our sites. None of them had very bad detection probabilities at all. And many of these were dual, in fact, all of these were dual arrays down here. So When we combined those, we got, I think, detection probabilities in the maybe at least, let's say, 80%, but probably more like 90 and above. So I think we just had low survival in that region.
So maybe I've got the wrong slide. I thought there was one slide where in I don't want to call it the North Delta, but where that 473 and where the 158 was, you had higher numbers than you had lower down.
No, here?
No, I must have been seeing things. Sorry about that.
Well, here we have more fish here than we had upstream, and that's because we had an additional release there, and so this is including that supplemental release. Okay. All right.
I guess one more quick question or a comment perhaps on your survivorship probability. I mean, a 0.5 survivorship probability sounds really high, except you're talking about fish that are almost 300 millimeters long. And so the other way of looking at that is that it's actually, I mean, if you look at Salmonid mortality patterns, it's actually pretty high mortality for a fish that big.
For a fish that large. Yeah, that's a very good point. I think having been used to looking at Chinook was very exciting, but yeah, working with a larger fish, maybe not as exciting. And comparing that to, say, the Fraser and the Columbia, where you would expect survival to be about, on that order of magnitude for much smaller fish and over a longer range, much longer than 91 kilometers. I think that's a good point. Okay, thank you.
Thank you.